Somewhere in your genome, there is a stretch about 10 million letters long. In it, something has been removed. You carry no Neanderthal DNA there, none at all. Neither does anybody else. Not one person tested, anywhere on Earth. That is strange, because if your ancestry runs outside Africa, about 2% of the rest of you came from them. 2% sounds like a rounding error. Your genome runs to 3 billion letters. 2% of that is 60 million. 60 million letters of you were written by another kind of human, one that has not existed for 40,000 years. And then these blank stretches where none of it survived. Something took it out. Something kept it out. Geneticists call them deserts. The biggest one holds a gene involved in speech. So what is still in you? What was stripped out? And what did the stripping? Here is the route. What 2% means, what the borrowed genes do, the deserts, the experiment that switched an archaic gene back on, what the tests cannot tell you, and the bill. Let's go. Start with the 2%. The number is stranger than it looks. In 2010, a team led by Svante Pääbo published a Neanderthal genome. It was read out of powdered bone. The comparison came back at once. People outside Africa share stretches with that bone. People inside Africa mostly do not. There is only one way that happens. Contact. Our species met theirs. They had children. Those children had children. Pääbo took a Nobel Prize for it in 2022, the first ever given for reading the genome of an extinct human. Before going further, be clear about who we are talking about. Neanderthals were not a rough draft of us. They were a species that ran for about 400,000 years. Their range went from Wales to southern Siberia. That is wider than most empires ever managed. And the brain case is not smaller than yours. Average volume runs near 1,450 cubic centimeters. Ours averages a little under that. Size is not the whole story, and nobody claims it is. But the cartoon is wrong. They made tools, pigment, cord, and fire. They cared for the injured. We opened one of their graves in an earlier video. Now, how anybody reads DNA out of a bone that old? This is the part that makes the whole field possible. DNA does not survive intact. It breaks apart. After 50,000 years, the fragments are tiny, often around 50 letters long. So you are not reading a book. You are reading confetti and rebuilding the page from it. And there is a worse problem. Modern DNA is everywhere. On the tools, on the hands, in the air of the room. A single flake of skin from an excavator can swamp the whole sample. The fix is elegant. Ancient DNA carries a specific chemical damage at the ends of every fragment. Cytosines decay in a way that leaves a signature. Modern contamination does not have it, so you read only the damaged pieces. The rot is what proves the sample is real. Now the part that gets skipped. Your 2% is not my 2%. Different people carry different fragments. My chromosome 3 holds a piece yours does not. Yours holds a piece mine does not. So line up thousands of living people. Start collecting the pieces. Joshua Akey and others did exactly that. They rebuilt large parts of the Neanderthal genome out of people walking around today. Estimates run to a fifth of it. Later methods push higher. Read that again. A fifth of an extinct species reassembled from the living. They are not gone in the way that word usually means. They are distributed. And we have one case where the mixing is not a statistic. It is a person. In a Siberian cave called Denisova, excavators found a fragment of bone. Just a splinter, a few centimeters long. It belonged to a girl of about 13. She lived roughly 90,000 years ago. Her genome came back with a result nobody expected. Her mother was Neanderthal. Her father was Denisovan. Not a distant mixture, a first-generation child of two different kinds of human. We found her on the first try. Out of a handful of sequenced individuals from that cave, one is a direct hybrid. Think about what that implies. If a random splinter lands on a first-generation child, this was not rare. And it did not only run one direction. Sequence the Neanderthal from the Altai, and you find something in her too. Modern human DNA. It arrived far earlier, something over 100,000 years ago. So an earlier group left Africa, met Neanderthals, left a trace in them. That group left almost nothing in us. Whoever they were, their line did not carry on. Read that again. There was an earlier attempt at leaving, and the only surviving record of it is inside somebody else's genome. Now the honest correction, because the simple version is wrong. You will hear that Africans have no Neanderthal DNA. That is not what the data says. Later work found small amounts across African populations, around 1/3%. It got there by people carrying it back in. Human beings have never stopped going both directions. So the map is not two clean groups. It is a gradient made by walking. Nothing in it ranks anybody. Keep that in your head. This subject attracts people who want it to mean something it does not. Now what the borrowed genes actually do. Here the pattern is sharp. Sort the archaic fragments by how much effect they have. The top of the list is dominated by one system, the immune system, over and over. There is a clean reason, and it is the center of this video. Think about what our species was doing 60,000 years ago. Walking out of Africa, into places it had never been. New plants, new animals, new water. And in that water, organisms that had been practicing on the locals for 400,000 years. Neanderthals had been in Europe and Western Asia that whole time. Their immune systems had met everything that lived there. Ours had not. Ours had been tested by Africa. So the two groups met and had children. Some of those children inherited local immune equipment. Those children did better. Their descendants are why the fragments are still here. Toll-like receptor genes show exactly this pattern. So does a cluster of antiviral genes called OAS. That is not a story about a superior group. It is a story about who had already been standing in that particular river. The clearest single example does not come from Neanderthals at all. It comes from Denisovans, the other archaic group, known mostly from that same cave. Stop on them for a moment, because their situation is absurd. We have a finger bone, a few teeth, a piece of skull, a jaw from a cave on the Tibetan plateau. That is close to the entire fossil record of a whole kind of human. You could hold all of it in 2 hands. We know more about their genome than about their faces. They were named from DNA, not from a skeleton. Nothing like that had ever happened before in the study of human origins. The gene is EPAS1. It changes how your body answers thin air. At altitude, there is less oxygen. The ordinary response is to make more red blood cells. That thickens the blood. Thick blood strains the heart. It makes hard work dangerous. It makes childbirth dangerous. The Denisovan version turns that response down. You adapt without the thickening. Emilia Huerta Sanchez and colleagues found it at very high frequency in Tibet. Around 81% of people there carry it. Now compare a European. The odds of carrying it are near 1 in 100,000. 81% against 1 in 100,000. That is not chance. That is a mountain keeping something on purpose. And the mountain did not evolve it in us. It was already there, in somebody else. We married into it. There is a second Denisovan inheritance, and a lot of people carry it. A region around a gene called TBX15 turns up at high frequency in Greenland Inuit populations. TBX15 is involved in how the body lays down fat and how it answers cold. Same shape as the last one. A local problem, already solved by somebody local, acquired by marriage. The same shape turns up in a virus nobody planned for. In 2020, Hugo Zeeberg and Svante Pabo found a stretch on chromosome 3. It raises the risk of severe illness from the new coronavirus. Carrying it was linked to roughly 60% higher odds of ending up in hospital. That stretch came from Neanderthals. Then they found a second one, chromosome 12. It goes the other way, same source, opposite effects. One encounter, both results. Now the part that keeps this honest, and it matters more than the genetics. 60% sounds enormous. Put it beside the social numbers from the same period. Being poor moved the risk further. So did a crowded job you could not walk away from. The gene is real. It is also not the biggest thing in the room. Anybody who tells you otherwise is selling something. You were not designed to leave Africa. Read that again because it is the center of this whole video. Nothing about the body that walked out was tuned for where it was going. Not for the altitude, not for the winters. Above all, not for the local organisms. Another kind of human had spent 400,000 years solving those problems. We did not evolve the answers, we acquired them. One generation at a time, by having children with the people who already had them. Now turn the whole thing over. The interesting part is not what stayed. The interesting part is what was removed. Line up thousands of genomes. Mark every place archaic DNA appears. Most of the genome comes out speckled. Fragments here, fragments there, different in every person. Then you hit regions where the marking stops. Wide, clean, empty. No living person carries archaic DNA there. The ancient skeletons we have sequenced do not carry it there either. Geneticists call these deserts, the biggest run to millions of letters. A desert is not an absence of evidence, it is evidence of a deletion. Because the mixing happened everywhere along the chromosome. The fragments started out scattered across all of it, so something removed them from these places. Fast. And from everyone. That something is selection. In those regions, the archaic version was worse. Now the two deserts worth knowing. The first is about 10 million letters long. Inside it sits FOXP2. People call FOXP2 the language gene. That is a bad name. It does have a real story. There is a family in Britain studied for decades where one copy does not work. Their comprehension is fine. Their intelligence is fine. What breaks is the fine motor sequencing of speech and the grammar that rides on it. So here's the obvious conclusion. Speech is what separated us. The archaic version failed. Selection cleaned it out. Obvious, tidy, and probably wrong. Because Neanderthal FOXP2 has been sequenced, it is nearly identical to ours. They had the same version. Whatever that desert is about, it is not a simple story about who could talk. That is the honest answer. It is more useful than the tidy one. The second desert sits on the X chromosome. This one tells you what happened inside those families. Archaic DNA is unusually scarce across the X. It is scarcest around genes that work in the testes. That pattern has a name in biology. It shows up whenever two lineages have been apart a long time. The sons of mixed pairings tend to have reduced fertility. The daughters are often fine. Now picture that on the ground. The crosses happened. Children were born and grew up. And a good share of the sons had few children, or none. Their particular fragments went nowhere. Two populations, apart for something like 500,000 years, meeting again at the exact edge of being unable to merge. Close enough that it worked. Far enough apart that it worked badly. The deserts are where it did not work at all. There is one more number that explains why the absorption went the direction it did. Neanderthals were never numerous. Estimates of their breeding population run in the low thousands across a continent. And their genomes show it. Long stretches with almost no variation. One individual from the Altai had parents who were close relatives. Half-siblings, on the best reading. Sequence a small population for long enough, and you see the same thing everywhere. Everyone is somebody's cousin. Now put a larger, growing population next to a small, thin one. Run it for a few thousand years. You do not need a conflict to explain the result. You need arithmetic. Some of you are thinking this is all marketing for spit tests. Fair question. Two things need separating. One is solid, one is not. The 2% is solid. It was found before consumer testing existed. Out of a bone, in a cave, by comparing sequences. The comparison is not a vibe. Specific variants in living people match specific positions in a 50,000-year-old skeleton. Now the oversold part. The trait reports. A report saying you carry more Neanderthal variants than 85% of users is a percentile among that company's customers. That is a ranking inside a shop. It is not a measurement of your body. And most single variants do very little alone. Think about how tall you are. Height is influenced by something like 12,000 genes. Each one nudges the number a fraction. Sleep timing works the same way. So does pain sensitivity. One archaic variant is one small voice in a very large room. So the ancestry is real, and the horoscope is not. Both are true at once. Now the second confirmation, because so far this is statistics run over databases. Statistics find associations. They cannot show you a mechanism. For that, you have to put the gene into something alive. Then watch. That has been done. The results are genuinely strange. Alison Muotri's lab took human stem cells. They edited one gene, NOVA1, back to the archaic version. Then they grew the cells into brain organoids, clusters of neural tissue in a dish, a few millimeters across. The archaic ones came out different surface shape, different synapse proteins, different patterns of firing. Now be careful with that. It is one laboratory and one gene. The paper has been publicly contested since it came out. Nobody grew a Neanderthal. Nobody came close. But the method is the point. Take a single archaic letter, put it back. Watch living cells answer. There is a cleaner case on another gene, the Neanderthal version of SCN9A. SCN9A builds a channel in your pain nerves. Put the archaic version into cells and measure it. The channel switches on at a lower threshold. That is not an association in a spreadsheet. That is a measured property of a protein. So people carrying it are not imagining anything. Their pain wiring trips earlier, and you can watch it happen on a bench. So, what ended it? The mixing ended, and the record of when is unusually sharp for something this old. Go in order. The main encounter dates to somewhere around 50 to 60,000 years ago, probably in Western Asia. Then it kept happening in smaller amounts as people spread out. The fragments in living Europeans and East Asians differ. Those differences record separate meetings. There is even a clock inside the DNA itself. Every generation, chromosomes get cut and reshuffled. So an inherited block gets shorter each time it is passed down. Long, archaic blocks mean a recent ancestor. Short, scattered ones mean a distant one. Measure the lengths, and you can date the encounter without a single bone. There is an older case still, from a cave in Czechia. A skull from a woman, sequenced in 2021, around 45,000 years old. Her Neanderthal segments are long. Long segments mean recent. The estimate puts the mixing 2,000 to 3,000 years before she was born. And her line went nowhere. She has no descendants among people alive today. So the picture is not one clean handover. It is many meetings, many small populations, and most of them running out. At a cave in Romania called Oaz, there is a jaw about 40,000 years old. Sequence it and the arithmetic is remarkable. That person had a Neanderthal ancestor 4 to 6 generations back. Not a distant echo, a great-great-grandparent. The last Neanderthal sites we can date run to roughly 40,000 years ago. Then nothing. Here's the part almost nobody mentions. The percentage has been falling ever since. Early modern humans in Europe carried more than we do. Some of the oldest sequenced individuals run to 3, 5, 6%. We are down to about 2. Selection has been quietly editing them out of us for 1,500 generations. The 2% is not what we inherited. It is what is left after the removal. Now the bill, in numbers you can check. Something over 40 million people have sent a sample to a consumer testing company. Most of them wanted a pie chart of countries. What they handed over was a genome. And a genome is not a password. You cannot change it. In 2025, one of the largest of those companies filed for bankruptcy protection in the United States, which raised a question most customers had never asked. When a company holding millions of genomes is sold, what exactly is being sold? And your genome is not only yours, it is most of your siblings, half of each parent, a quarter of your cousins. None of them spat in a tube. All of them are in the database now. That is the modern trade, and it is worth seeing plainly. We turned ancestry into a retail product. 60 million letters arrived in you across an encounter that lasted thousands of years and left almost no other record. You can now look them up for the price of a takeaway. The cost is a permanent copy of your family, held by somebody else. So what if the 2% was never the interesting number? What if the record worth reading is the empty part? The deserts. Every fragment that survived only tells you it was harmless or useful. The blank regions tell you something else. They tell you where a whole species was incompatible with us. And what if that is the honest description of what happened? Not a conquest, not a merger, an absorption, with an edit afterwards. An edit that has run for 1,500 generations and has not finished. We traded a second kind of human for 2% of their genome, and most of us never even knew it was gone. So go back to that stretch of 10 million letters. It is in you right now. It is completely clean. And it is clean in every person on the planet. Whatever sat there did not work in a body like yours. Something removed it before there was anybody to write it down. That is the only obituary they got. It is written inside you. Now I want to hear from you. 2 questions, and the second one is the real one. First, the X chromosome desert. 2 species or 1 species drifted too far apart? Pick a side. And second, have you sent your DNA to a company, knowing it is also your sister's and your father's? I suspect this comment section splits hard, and I suspect it splits by age.